Are you anxious to see what a tree job returns? Let's go to our workspace for our jobs page to look at it. So you can click on workspaces and my jobs, or of course you could navigate to it, but I'll click on my jobs. And let's start with this one, the reference and public and the ANL 2025 genes. So I click on that and I click on the view icon. Now, in this particular video, I'm going to show you a lot of the stuff that comes with this job, but not the viewer. We'll do the viewer next. So you can see that it took 1 hour and 19 minutes to load. And there are all these files here, so many different files. It's just showing you everything that they have here. And if you wanted to take the alignment and do something with it, all of that stuff is here for you. You could view the tree this way, or with the file XML, view it here, but we're saving this because it's going to be the big reveal. I don't want to have a spoiler in this video that ruins the next one for you. And also there are the detail files. So there's like so much stuff that comes with this. So what does one look at? The thing I always look at first is this, the report HTML. And some of these other things, it's just a list of the data that was used. And you might want that, like if a reviewer specifically asks for it. But this is for you, and it informs you about your tree and what was the data used to generate it. So I click on that, and I click on the view icon. And it shows us a tree, a static view of a tree here with all of the genomes. You can't see any of the groups. You can't cover it right now. But look, it's got a really nice way of showing you the sort of outgroup genomes. Spoiler alert, these are the reference genomes. And so it makes the tree, I think, look nice. And then you come down to the tree analysis statistics. 95 requested genomes, 95 genomes had data. 100 genes were requested, 100 single copy genes were found, and then it gives you the number of protein alignments. And this is the part here that I start using for publication. It was 40,444 aligned amino acids. 100 CDS alignments because there were 100 genes, and then 121,332 nucleotides. So that's a lot of data going into a tree. I think that should produce a well-supported tree. If you care about command line for XML, there it is. It also tells you the codon partitions, which we talked about in the introductory. Now, it gives you some warnings, right? Some of these things are exactly identical, and you may be thinking, what? There were identical genomes in there? Remember, this isn't looking at the entire genome. It's looking at 100 protein families off of a genome that has, I think strep has like 1,500 genes across the whole thing. So in those 100 genes, it looked at those and said, gosh, for those 100 genes, these guys are identical. It means they're probably really close genomes, but are they the exact same thing? I don't know that I could say that. And if you were interested, you could copy this and do a find and find out where it is in this thing. So you could do that if you were interested. Oh, see, 1,500 genes. So it tells you what the genomes were with their genome ID, how many single copy genes they had. They only used 100 of those. So you could make a bigger tree if one wanted to, meaning you could add more data, but it would take more time. And let's just scroll down a little bit farther. And then it's looking at the protein families that were used, and you could download that information too. And that just shows you what those protein families were. And if you look at the top, it tells the alignment length. So it gives you the information on those. And if we scroll down, that should be it for the report. So for this particular video, I just want to really concentrate on this report. There are a lot of files there if you want to look at it. In the next video, we're going to dig into the special viewer that BV-BRC has for the trees. So we'll start that next. And that is really going to be awesome. So stick around.